Good evening, everyone. How are you? Okay, excellent. Thank you for coming. My name is Caleb. I am a student with the University of Maryland. I'm also the president of the university's student chapter of the American Society for Biochemistry and Molecular Biology. I would very quickly like to thank the ASBMB members, the Rockville Science Center, the university staff, and tonight's presenter for their effortless collaboration in bringing this event to you. Tonight, we have a very, very special guest, Dr. Daniel Nelson. He's an exquisite scientist. He is the head of the Laboratory for Antimicrobial Discovery for the University of Maryland. He contributes within the Department of Veterinary Medicine of UMD and the Institute for Bioscience and Biotechnology Research. And as if that was not enough, he also is an associate professor at the University of Maryland. Additionally, he consults with various study sections of the National Institute of Health, NIH. He also collaborates with both private and public companies who are developing antimicrobial technologies. And the coolest thing of all that Dr. Daniel Nelson does, the coolest thing is the fact that he is a subject matter expert on antibiotic-resistance for the Department of Defense. So that means when the U.S. government, the Department of Defense, needs an expert about antibiotic resistance, they come to this guy. Which brings us to tonight's topic: antibiotic resistance, antibiotics, and much, much more, brought to you by Dr. Daniel Nelson. So, let us wait no longer, and join me in welcoming Dr. Daniel Nelson.
Thank you. All right. Can everyone hear me okay? Okay, well, I want to thank Caleb and the rest of the students for inviting me. And by the way, there are tons of subject matter experts at the DoD. It's not that high of a bar. But I'm here today to talk about antibiotic-resistant bacteria. I actually gave a version of this talk to this same group 4 years ago, so presumably most, if not all of you, were not present at it, which is why I was asked to come back again. And the format was a little different. Back then it was a little more discussion. I wasn't allowed to use slides. I just sat here and we had an open discussion. And I actually like that format. But giving slides, there are some advantages. There's some neat flash animation and movies and videos and graphics that I can show. But even though I'm using slides, I would like to treat it as if this were just a discussion and I was sitting here in this chair without slides. Meaning, please feel free to interrupt me, stop me. If we don't get to all my slides, that's fine. The whole point of this is to have a discussion. And this is geared, this is by the undergraduate students here. But it's geared for middle school students, high school students, and undergrads. So it's not going to be too technical. It's going to be more of an overview, but there will be some technical aspects. So again, please ask questions if you have any. And the order I'm going to present, normally I would talk about myself and how I came to be where I am and what grad school was to me, and then I give the talk about the antibiotic-resistant bacteria. But I'm going to do it in reverse. There was a speaker last week, and it worked out well that way. She presented the research component, and then she spoke about herself. And in general, it seems like most of the people in the audience are interested in that latter half. Why did you go to graduate school? What is graduate school versus medical school? How long did it take you? What about getting in? What are the costs? It seems many questions as you may have about antibiotic-resistant bacteria, those seem to be the questions that a lot of the students have. So I'm going to actually end with my personal journey and background, but I'm going to go right into the antibiotic-resistant bacteria talk. One thing, if you ask me a question, I'm going to repeat it because the only live microphone is the one right here on me, and for the people we're streaming this to, that's the only way they'll be able to hear. So ask questions, but then don't be offended if I repeat your question. So we've all seen headlines about superbugs, a crisis, pandemics. Is this real? Is this hype? That's kind of what we're here today to discuss. And I'm not over the hype myself. In fact, The University of Maryland last year had this on most of their bus stops down in College Park. I actually went to our PR people to talk about some work we were doing with Streptococcus, a bacterium, and they said, that's neat. What else do you work on? I said, well, I work on methicillin-resistant Staph aureus. They're like, that's even neater. That's more scary. What else do you work with? I'm like, well, I haven't worked on it in a while, but we have some projects on anthrax. Immediately, there are headlines, anthrax. And in fact, in this picture here, I'm holding a blank dish, so that's all computer simulated. This was the hottest day of the year up in an attic under the bright lights. I was holding this thing for 2 hours, so my gloved hand was completely full of sweat, and they photoshopped the sweat out. And in fact, I was wearing a light color shirt, and it didn't have enough contrast. So everything about this is more or less fake, other than the fact that I really do have some work on Bacillus anthracis, even though we haven't done it in many years. So there is a lot of hype, and you need to be able to look through it, yet some of it is real. There is concern about the growing antibiotic crisis that you keep hearing about. So let's back up a couple of steps and start very basic. What are bacteria? The first thing is that bacteria are not viruses. This is the biggest thing the media gets wrong. They talk about the HIV bacteria, well, it's a virus. Or they talk about the Staphylococcus virus, it's a bacteria. So the first thing that bacteria are is they are not viruses. They're very, very different. Antibiotics do not work on viruses at all. They only work on bacteria. So getting to the bacteria, they're single-cell organisms, and they're not all that different than us, actually. They have genes. Their genes are made of DNA. The DNA becomes RNA. The RNA becomes proteins. Those proteins help the cell function. They need food. They need for energy. They need nutrition. They break down that food. They have metabolites. They have waste. They have ways to protect themselves in these outer membranes and a bacterial cell wall. And they even have pseudo legs. They have a way to move around. So a bacterium really isn't all that much different than we are. A lot of the same processes. So we use antibiotics to try to kill them, but that gets a little tricky because, as I said, we're very similar to bacteria. So how do we do this? So I'm not a big fan of using Wikipedia as props, but I did find this on Wikipedia under the antibiotic section, and it does a pretty good job explaining it. So here's a bacterial cell, and cells have membranes, just like all of us. Each cell in our body has a plasma membrane. Bacteria have a plasma membrane, and then they also have a cell wall. This cell wall is a rigid structure that keeps the bacteria together. So antibiotics can act on this cell wall, which is unique to bacteria. We don't have cell walls in us. And so some of the most basic, the earliest antibiotics, penicillin, the first antibiotic, acts on the bacterial cell wall. But some of the most sophisticated and advanced antibiotics, the glycopeptides, also act on the cell wall. You can have antibiotics that act on the membrane. Polymyxin B, colistin, these are antibiotics that act on the bacterial membrane, but because we have membranes, there are some issues with toxicity, but they do work very well on some bacteria. Again, the bacteria have DNA, RNA, and they have ribosomes where they make proteins. So there are different antibiotics that act on the DNA. You have the fluoroquinolones that act on the enzymes that replicate the DNA or unwind it. You have enzymes, or you have antibiotics that act on some of the enzymes involved in the translation of DNA to RNA, or the transcription of RNA to DNA. And then you have enzymes that bind to the ribosomes and prevent proteins from being made. Just the basic cellular processes of life. And then bacteria also need certain metabolites, certain chemicals, in this case folic acid biosynthesis are required, and so antibiotics can act on enzymes in these required metabolic pathways. So when they talk about classes of antibiotics, the different classes act on one of these 5 or 6 different areas. And then there are different generations of antibiotics within each class. So let's take the first discovered one, penicillin. So penicillin was discovered by Alexander Fleming in 1928. And for many of you may have seen his picture. Does anyone know how it was discovered? Exactly. He made this discovery by accident. It was actually a contamination. So he had a petri dish full of his bacteria, and there was a piece of mold, a common bread mold, the penicillium. And around that mold, there was this clearing zone. And so intuition said, well, something in this mold is secreting something that's obviously killing the bacteria. So let's grow up some of this mold, filter out this substance, and see if it kills, and sure enough it did. So it was an accidental discovery that he made in the laboratory in 1928, worked on it for many years, and the first use of penicillin took place in 1942. It was in England. The patient was Anne Miller. She was in her late 20s. She actually went through childbirth and had a bad infection and was about to die, and they decided she would be the first person to get penicillin. At that point in time, the amount of penicillin they gave her was half of the world's supply, because this fungus produces very small amounts of it, right? So they used half the world's supply of penicillin on her, and it saved her life, and she did not die until the 1980s. She was well into her 90s, 95 or 96 years old. So that was the first use of penicillin, and so many people credit that this begins the antibiotic era. This is when it starts, this discovery, or this first use in a human saving someone. So by the end of 1942, they ramped up production, and they had enough penicillin to treat 10 patients. In the entire world, this is. 10 patients in the entire world, and it worked on all of them. But what was happening in 1942, 1943? It was World War II. So in the war, you had more people dying from infection than from being shot. They may be shot, they may get a blast injury, or they may not. They had, it was a trench foot just in the trenches with poor sanitation and not enough warm clothes or hygiene. They would get infections in their skin and they would die, and there were no antibiotics. So there's a huge war effort here in the US and in England and other parts of Europe to ramp up production, such that by the time D-Day occurred in 1944, we went from 10 doses to 2.3 million doses of penicillin. By 1945, they were able to produce 600 billion units of penicillin per year and save tens, if not hundreds of thousands of lives during the war, and also in civilian populations by that point in time. In 1945, Alexander Fleming won the Nobel Prize for his work on penicillin. And shortly afterwards, let's see at the bottom there, shortly afterwards in a speech, he said, the thoughtless person playing with penicillin treatment is morally responsible for the death of the man who succumbs to infection with, I'm not sure why we can't see it, with the penicillin-resistant organism. So that was some pretty forward thinking. Here he is, the inventor of penicillin, or the discoverer, winning the Nobel Prize, saying, we need to be really careful, there it is, we need to be really careful about this, because resistance, if we aren't careful, resistance is going to be a huge issue. So was he just brilliant? Was he a fortune teller? Could he see into the future? No? He was just good at reading the literature, because in 1940 there were already publications of enzymes that could destroy penicillin. So they called them penicillinases, and they were able to destroy penicillin and render the bacteria resistant to it. So that occurred in 1940, before the first human use of penicillin. So I would argue that if you say 1942 begins the antibiotic era, it was actually over before it began. There was resistance before it was ever used on a single human being. So this is kind of a scary graph. What this shows on the top is when different antibiotics were first used on humans, and on the bottom is when resistance was developed. So going back to penicillin, we see here its first use was 1942. Now in the laboratory, it was already resistant in 1940, but it didn't start showing up in humans and hospitals, resistant isolates, until about 1946. You go through this chart, you can see tetracycline introduced in the late 40s. By the early 50s, there was already resistance. Methicillin introduced in 1960. By 1962, you had methicillin resistant. Cephalosporins, you had resistance almost immediately. You come down here to more modern times, these are the, the fourth generation, or these are the latest frontline antibiotics we have. Linezolid approved in 2000. By 2003, there were resistant isolates. Daptomycin, the same story. So the time frame is getting even shorter. So that's when people talk about the antibiotic crisis, this is exactly what they're talking about. So not only do we have emergence of new resistance, the incidence of resistance is increasing. If we can actually go backwards here, methicillin is a very powerful antibiotic. It was introduced around 60. Resistance was noted in 62 or 63, but no one cared. This was extremely rare, 1 in a million people, right? Very rare. In fact, no one cared about methicillin resistance until probably 1990 or 95. The reason being, it was so rare we just didn't see it. Here's the incidence of methicillin resistance. It was down around 1% or so in 1980. But come 1995, all of a sudden it's 35, 40%. Around 2008, it's up around 60%. Now it's around 70%. So 70% of all, this happens to be Staph aureus, 70% of all Staph aureus is resistant to methicillin.
Is there in the graph, it seems that it took the longest for there to be erythromycin resistance? Is there something special about erythromycin, or just luck?
It just, the way the genes are transferred between the bacteria. So we'll talk about it a little bit later, but how do you treat methicillin-resistant Staph aureus? One of the last line of defense is an antibiotic called vancomycin. Guess what? 10 years ago, there became vancomycin-resistant staph, and everyone thought, oh my goodness, in 5 years it's going to be everywhere. Well, you know what? 10 years later, there's only been 13 cases ever reported. So it's out there, but we expected it to take off, and it just hasn't. That doesn't mean it won't. It probably will, but you don't know, just the way the different genes are transmitted. So this is methicillin-resistant Staph aureus, or MRSA. We're going to talk a lot about this later on in the presentation. These are vancomycin-resistant Enterococcus and fluoroquinolone-resistant Pseudomonas. So not only are bacteria becoming resistant to antibiotics, but the percentage, the incidence of that resistance is dramatically increasing. So let's look at a drug development timeline. Whether it's a drug for diabetes or an antibiotic or for cancer, how much does it cost to get a drug approved here in the United States? A lot. Try $500 million to $1 billion. And how long does it take? Years and years and years. So this is a chart showing, and most of what we do in academia is down here around drug discovery or preclinical. This is where you're just showing that it works, how well it works. You're doing some basic animal studies to show that it has protection. Once you start to get to the pharmaceutical companies, that's where your money and time really ramps up, and you go through the different phase 1, phase 2, phase 3 safety trials, large-scale trials. This is what starts costing hundreds and hundreds of millions of dollars. So with that said, I need a pharmaceutical company CEO. Who wants to volunteer? Someone does. Joshua, get up here. Joshua is a 7th grader, and he actually did a science fair project, worked in my lab 2 years ago. But he doesn't know what I'm going to do now, so I didn't plant this question. Sit down right here, Joshua. Okay. So you are a pharmaceutical company CEO. Your base salary is $5 million a year, but that's nothing because your bonus last year was $20 million. And then your stock options were another $20 million. So you made about $45 million last year. You run a very large company, and you have to talk to your shareholders, and you have to please them. And they're expecting you to come up with the next blockbuster drug. So you've got $1 billion, but they want you to turn this $1 billion into a drug that makes $5 billion a year. So your scientists come to you, and they start pitching drug A. This is good. You can't see what's behind them. They start pitching drug A, okay? Drug A is something that works on a chronic condition. Chronic condition meaning you take it every day. And I'll give you some examples. I have high blood pressure. I take a blood pressure pill every day, 365 days a year. I've been doing it for 10 years. I'm probably going to do it for the next 40 years or however long I live. Someone's making a lot of money off of that. My wife, where's my wife? She has high cholesterol, so she takes Lipitor to lower her cholesterol. She takes it every single day, 7 days a week, 52 weeks a year, most likely for the rest of her life. Someone's making a lot of money off of that. Arthritis, diabetes, asthma, depression. These are things that people take drugs every day chronically, okay? There are also unique treatments. So not everyone has cancer, but there are certain cancer drugs for certain types of cancer that they're the only treatment available. So you can charge a lot more money for that, even though not everyone takes it, right? And you have long patent lives on these drugs. Okay, so that's the scientist that tries to tell you about drug A that treats a chronic condition. Then another scientist, then I come in, and I say, let's talk about drug B. It's an antibiotic. Now, you only need it every few years. So who has taken an antibiotic in the past year? Okay, in this entire room, we've got 4 people, 5 people, okay? For some of the adults, how many antibiotics have you taken, say, the past decade? I think I've taken twice, okay? Okay, so this is a drug you're only going to need every couple of years, okay? And you take it for a week or less. If you take a Z-Pack, you take it for 3 days, okay? There are many competitors that sell many drugs in the same class, and the largest need may actually be in underdeveloped countries that you have to sell it for pennies a dose. And the thing with antibiotics is the more you use it, the less effective it becomes. And oh yeah, by the way, you remember that slide before where I say you go through all this money expense and it gets approval, and then a year or two later there's already resistance? Okay, so now that's my drug B, the antibiotic. So now you are this pharmaceutical CEO, and you're going to take your $1 billion. Are you going with drug A or drug B? Drug A, I thought so. Thank you, Joshua. And in fact, that is, unfortunately, what is happening. So drug companies are getting out of the antibiotic business. Here's another thing I took from Wikipedia this morning. These are the largest selling pharmaceutical products. Over here you can see the amounts. That's $5 billion a year, up to $14 billion a year. You look at them, they work on rheumatoid arthritis, Crohn's disease, here lymphomas, cancers, diabetes, cancers, asthma, cardiovascular disease. There's your Lipitor. So these are chronic problems people take every day, or they're highly specialized cancer drugs. You have to go very far down that list to find an antibiotic. And so because of that, drug companies are getting out of the antibiotic business. This shows, so when you apply for a new drug to the FDA, it's called an NDA, for a new drug application. These are the new drug applications over the past 20 or 30 years. Now, this is a little bit cheating because this ends here in 2012, and actually there are figures now out here where the level has bumped up to 5 or 6. So the trend's starting to get a little better, but in general, you know, the fact that pharmaceutical and biotech companies, by and large, not all of them, but by and large, many of them are drawing back from this line of research. So here's a quote from, this is a few years old, 2013, I believe, from the Assistant Director of the Centers for Disease Control. And he said, we're in a post-antibiotic era. There are patients for whom we have no therapy and are literally in a position of having a patient in a bed who has an infection, something that 5 years ago even we could have treated, but now we can't. So that's what it's getting to. Now, we're not talking thousands and thousands of patients. You know, the Shady Grove, hospital doesn't have hundreds of patients with infections that can't be treated, but the trend is worrisome. So what I'm going to present in the next several slides were all taken from this brochure. It's Antibiotic-Resistant Threats in the United States. It was published by the CDC in 2013. It has not been updated since then, but all the information in it is still fairly valid. It's just a few years old. You can go online, just Google it. It's like a 120-page PDF. You can download and look at the whole thing. does a nice job talking about antibiotic resistance. How does it happen? So let's start with germs. Okay, say germs in your gut. The blue ones are not antibiotic resistant. Maybe they're good, healthy, happy germs. And the pink ones here are antibiotic resistance. When you take antibiotics, you kill hopefully the bacteria causing the illness, but you also kill some of your good bacteria, known as your microflora. And what's left are these antibiotic resistant bacteria, who all of a sudden now start to grow and multiply. And so that can be an issue. Additionally, they can start to transfer their antibiotic resistant genes to bacteria that don't have antibiotic resistance. So that's how the resistance happens. And there's an important term that we need to learn here. Does anyone know what nosocomial means? Same person. Yes? Chris. Hospital acquired. You hit the words. So it means hospital acquired. So this becomes a problem when grandma falls and breaks her hip and goes to the hospital and has a hip replacement and is just fine. The hip's starting to heal, sutures look great, and then she has a horrible multi-drug resistant infection. Okay? That's a hospital acquired infection. Now, that can come from within, so it can be right here. Because we don't want grandma to get sick, we put her on tons of antibiotics, we kill all the good bacteria, and then the few bad bacteria that are held in check by the good bacteria can now grow and proliferate. So she could actually get the infection from herself. She may already, we all have, for example, C. difficile in our guts. Pathogenic C. difficile is just held in check. But you go on powerful antibiotics, kill everything else, and then you get this differential growth of the C. difficile, or she can get it from the hospital itself. Look at all these places in a hospital, all the linens, all the devices, the suctioning, the blood pressure cuffs. It's just crawling with bacteria. And so people who are sick, there is a push as soon as they're better to get them out of the hospital because you don't want them to catch an infection. So this is also in that booklet, and this talks about how the resistance spreads. So George here gets antibiotics, and he develops resistant bacteria in his gut. Now he can go to the hospital, and he can spread it through the hospital to inanimate objects, through different medical devices, directly to doctors and nurses who bring it home and spread it to their family, or he can stay and spread it to the community. So humans are one side of the spread. The other side is agriculture, is farm animals. We're going to get to this later on, but they're giving loads. 80% of all antibiotics are given to farm animals, not humans. And there's reasons for that, good and bad. But 80% of every antibiotic that's produced in the world goes to a farm animal, not a human. So they get these antibiotics, they develop resistant bacteria in their gut, and the antibiotics can survive the food processing and we consume it, or in the wastewater that comes off of the farms, it gets into the groundwater, it gets on crops, and we can consume it that way. Either way, the animals and humans, we both spread it all together. So there's this concept called One Health, where the NIH, which is human disease, and the USDA, which is animal disease, they're finally saying, you know what, we aren't different. We're actually, it's one global health, animals and humans, and we need to start thinking about it that way. Okay, so I'm going to go over just some of the more common bacteria. And so in this report, the CDC labeled bacteria as an urgent threat, a severe threat, or an emerging threat. So at the top of the list, the urgent threats, and I've not pulled out every single one, I've just pulled out a few representative examples that some people may have heard of, is C. difficile or Clostridium difficile. Again, this is an organism probably every one of us here already has in our intestines, but it's held in check by our good bacteria. This is the one when you go to the hospital or go on massive amounts of antibiotics, you can get severe C. difficile infections, over $1 billion a year in excess medical costs, 250,000 infections a year, 14,000 deaths, but if you read through here, there's a drug-resistant strain, and it's led to a 400% increase between 2000 and 2007, just due to this drug-resistant strain. So that's C. difficile. And then there's carbapenem-resistant enterobacteriae. So carbapenem is a strong antibiotic, and the enterobacteriae, this is Klebsiella or E. coli. There are 9,000 infections a year, 600 deaths, and it's split between Klebsiella and E. coli. And so sometimes this is referred to as CRE, C-R-E, for carbapenem-resistant enterobacteria, resistant to nearly all available antibiotics. But I'm going to tell you a special story now about one from Klebsiella. And they, just to confuse you and change up the name, this one they call KPC. The name of the bacteria is Klebsiella pneumoniae carbapenemase, because this bacteria secretes an enzyme, carbapenemase, that destroys carbapenem, which leaves the bacteria resistant. So CRE and CPK are essentially the same thing. And this story happens, this is my clue to go to the website. This was an outbreak at the National Institutes of Health a couple of years ago. And so what I'm going to show you, this is a PBS documentary on Frontline, hunting the nightmare bacteria. You can Google it. It's an hour-long show. You can download and watch the entire hour. What I'm going to show you now is the first 3.5 minutes of this program, and then we will chat about it.
This superbug didn't come from overseas. This one was homegrown. It lives in the digestive system. And like NDM1, it's a gene that can spread its resistance to other bacteria. It's called KPC. No one knows exactly how many patients in the New York City area have been infected with KPC, or how many have died from it. Nationally, most hospitals aren't required to report outbreaks to the government, and most won't talk publicly about them. But as part of Frontline's investigation, one of the nation's most prestigious hospitals, the Clinical Center at the National Institutes of Health, the NIH, agreed to recount how it dealt with a major KPC outbreak. It began in the summer of 2011, when a woman carrying KPC was transferred from a New York City hospital here to the NIH in Bethesda, Maryland.
Talking about hospital infections is really difficult for a hospital, because what you are saying is that we all know that when you come to the hospital, there are certain risks. But we've now laid bare what are those risks.
The NIH had never treated a case of KPC before. And as the patient was brought into the ICU, the staff was determined to keep the KPC from spreading to other patients.
We immediately went on high alert, the equivalent of hospital epidemiology DEFCON 5. Tried to implement as many things as we could think of at the time to prevent any further spread of the organism in the hospital.
They called it KPC. And so we learned later that was Klebsiella pneumoniae carbapenemase, and that's a mouthful. But we really didn't know what that meant.
The patient was placed in what we call enhanced contact isolation, which means everybody who went in the room, including visitors, had to wear gloves and gowns.
The room was at the end of the hall, separate from other patients. But this was the intensive care unit, where patients are very sick and highly vulnerable. and that presented heightened risks.
It's the kind of place where the bacteria can spread with ease. People are very busy and there are a lot of things going on. Patients get very sick very quickly and require intervention. The bacteria can be spread on the hands. They can be spread on pieces of equipment that might go from patient to patient, so you have to be really cautious.
Their efforts to contain the KPC appeared to work. When other ICU patients were tested for KPC,
we found nothing. So at that point, we thought that there had not been spread of the bacteria.
the New York patient ultimately recovered and was discharged after 4 weeks in the hospital.
We really felt like we had dodged the bullet.
But
But, and then it cut off. Okay, so again, this is Frontline. If you go to the website, you can download the entire thing, but they have this really neat flash animation thing here that tells the story. Patient 0 was this 43-year-old woman who came from New York City Hospital. They were on, she had KPC, they were on high alert. My wife works at the clinical center as a nurse practitioner at NIH, and she said, you were forbid to go anywhere near the patient unless you had some direct patient care. I believe they actually posted security guards on the ward that you couldn't go in and out, and they had to physically see you gowning up and washing your hands, so they went on very high precaution and sterile hygiene procedures. As this video ended, the patient later discharged, and the hospital officials believed that they contained the bug. And then a second infection. Several weeks later, a 34-year-old man hospitalized with a tumor tested positive for KPC. The two patients had no known link. They spent no time in the ICU together. They had different nurses and doctors, completely different equipment. And then the outbreak begins. Over the next few months, several more patients test positive for KPC. Then NIH attempts to deal with the outbreak, but no combination of antibiotics was able to stop it. Beyond the ICU, the hospital creates separate ICU just for these patients. The old ICU is cleaned and disinfected by robots. I don't know how they did that, but sounds neat. Before patients were moved back in, but somehow it spread beyond the ICU. Doctors tried several combinations of antibiotics, even an experimental drug that wasn't even approved yet, and nothing worked, and then the patients started dying. 5 other patients died, but the hospital believes their deaths are due to underlying problems, not the KPC. Ultimately, 11 infected patients dies. Looking for a link. So this is where epidemiology comes in. So as a hospital epidemiologist, you can look for the obvious. Well, this patient was in this room, and another patient in that room a few weeks later got sick. Or these patients were together in the same ward. Or these patients had the same set of nurses, right? And so that's a physical finding. So if you follow this, a black line, if you look on the right, at the patients, a black line means the infected patients were in the same ward, which likely provided the opportunity for the bacteria to spread. The brown line indicates the 2 patients had no overlap, but genetic analysis makes the link. So you can have the physical link, they shared the same ward, the same nurse, used the same wheelchair, whatever, but then you isolate the bacteria from them, and bacteria are continually changing and mutating. So if you sequence the entire bacterial genome, you can look for tiny little places where the bacteria changes, and if you add it all together, you can say, okay, this was the bacteria we started with, and this one has 10 changes, and this one only has 5, so probably this was patient 2 and this was patient 3. So you can do a genetic link, a genetic epidemiological link. So the brown lines indicate that the 2 patients had no overlap, but genetic analysis makes clear that the bacteria in the 2 patients were closely related and likely separated or came together, spread by a more complicated path. And then the dotted lines mean there's just other factors involved. There is a paper, let me go up here, if you go to this website, gathered from their own research. And so here is, you can download it for free. the PDF. And keep in mind, this could only happen in NIH. There's no Shady Grove Hospital or Suburban or any other hospital in America that has the resources to go through this type of epidemiology, to sequence completely every single bacterial strain, do the genetic analysis to the level they did here. This could have only happened at NIH, the only place in the entire world this could have happened. If you're so interested, you could read this paper and go through all of the graphs and figures and genetic analysis. Here's where they put the blueprint together. These are pretty small. I won't zoom in, but you can go there, and they looked at what wards they were in and what beds and who they were with. This is the genetic analysis of the different genes that had changed, and go on, so on and so forth. So in the end, find on page sync. Okay. So, however, I'll read to you what it says. The possibility of transmission through inanimate objects could not be ruled out and was in fact bolstered by culturing the outbreak strain from 6 sink drains and from a ventilator. The ventilator had been thoroughly cleaned after being used on one of the patients, twice with quaternary ammonium compounds, which is a strong disinfectant, and once with bleach. The ventilator isolated, distinguished it from the original isolate, confirming the isolate likely survived the cleaning process. All other environmental cultures were negative, with the exception of those taken from the room of patient 8 before decontamination. So even after these things were decontaminated, even after this ventilator was cleaned with strong disinfectants and with bleach, it still had the bacteria and was subsequently used on another patient. My wife's dying to ask a question.
So, do you know if they ultimately found out, was basically the source?
I think it's the ventilator.
It was the bottom of a handrail on the 5th floor, back corner by the lumber room. And on the wooden handrail that patients would use to walk, on the bottom of that handrail. So they went through the entire unit, they cultured every square inch of every wall and found it on the base of that handrail.
So there you have it. Even after that disinfection, they still could not get rid of it. And then to go back here, to finish this up, patient 0, the victims, looking for a link, one more patient. Then a year after the outbreak, a young man arrives at NIH due to complications from a bone marrow transplant. He becomes infected with KPC and dies. This is a year later. So today, NIH knows that KPC isn't gone. It's always going to be there, and it's always going to be an issue that they're going to face. So that was the KPC story at the most prestigious hospital with the most stringent epidemiology and disinfection practices. You have this massive outbreak, so it can happen anywhere. Yes?
How could the bacteria survive for a year without a host? Doesn't bacteria host?
No. No, they can go dormant. Not this particular bacteria, but other bacteria can form spores, live for hundreds of years. They can grow in a biofilm, and a biofilm is where bacteria shut down metabolically. Think of like a bear hibernating, so they don't need nutrition or anything. And in the biofilm, the thick biofilm protects them from antibiotics, and it protects them from disinfectants. So there certainly are ways and mechanisms which it can survive. Yes?
So if this was resistant to all known antibiotics, is that why the patients died?
Yes. And these were, keep in mind, these were pretty sick patients. Some of these people have gone through lung transplants and bone marrow transplants. So, these weren't healthy people who showed up with this infection. There were other, what they say, comorbidities involved. But yes, they did all end up dying, and none of the antibiotics could help.
So basically, you try to boost the person's innate immunity?
You can give them IgIV. You can do supportive care and boost other things where you can. but a lot of times these patients are already somewhat immunocompromised. So that's actually going to lead into a discussion in just a minute when we talk about strep. So just to finish up some of the bacteria here, I'll go through them quickly, but you might hear of ESBL. This is extended spectrum beta-lactamase producing bacteria. So they can produce enzymes that cleave the latest generation beta-lactam bacteria. You see here 1,700 deaths per year in the these figures are all the U.S. only. 140,000 infections, 26,000 resistant, 1,700 deaths. This is labeled as a serious threat by the CDC. Drug-resistant Campylobacter, 1.3 million infections a year, resulting in 13,000 hospitalizations and 120 deaths. And then what you can see, I don't know how well you can see here, but this is the incidence of resistance. So it's steadily growing, the number of resistant organisms. Salmonella, again, 1.2 million infections a year, 100,000 of which are resistant and climbing. Drug-resistant strep pneumonia. So there is a vaccine for pneumococcus approved for elderly people and young people. I don't know if some of you are young enough here that perhaps when you were babies you had a pneumococcal vaccine. It's a great vaccine. It saved a lot of lives. But the bacteria does change, come up with new serovars that are not covered by the vaccine, and most of us middle-aged people have never been vaccinated for it. 1.2 million drug-resistant infections a year, leading to 7,000 deaths. Again, just in the U.S. Drug-resistant tuberculosis. 1 billion people have tuberculosis. 1 out of 3 people in the entire globe have some form of tuberculosis, whether active or latent. Here in the U.S., it's not that big of an issue. There are only 10,000 cases, but 1,000, 10% are drug resistant, and around the globe they have some very strong multi-drug resistant strains that are resistant to everything. Again, this is labeled as a serious health threat. Group A strep, has anyone had strep throat? Okay, so group A strep is something I work on in my lab. And it's still pretty easy to kill it. It's one of the few that still has some probably the only one that still has some sensitivity to penicillin, but it is developing resistant to other antimicrobials such as erythromycin. Clindamycin-resistant group B strep. Women who are pregnant in the 38th week of pregnancy are often tested, or not often, I think they're always tested, it's a law, for group B strep, because during childbirth, as the child is coming down the birth canal, the infection can spread to the child and the child can get bacterial meningitis. So group B, and of course if you have the infection, women who are 38 weeks pregnant, the last thing they want to do is start taking heavy doses of antibiotics right before they give birth. Now we have multi-drug resistant group B strep to compound the problem. So this does lead to 27,000 severe cases a year with 440 deaths. And then we get to staph, favorite one of study. This is the favorite one that you see in most of the headlines. So Staph aureus is the name of the bacteria. It's resistant to methicillin. Methicillin is an antibiotic that no one's used for 30 years, but the name sticks because it defines an entire class of antibiotics. So it's resistant to this entire class of beta-lactam antibiotics, and so it's called MRSA, or MRSA for short. That's methicillin-resistant Staph aureus. It is a major healthcare-associated infection. So we talked about nosocomial infections that you get from hospitals. This is one of the major ones you get. And just about the only thing you can do to treat it is use vancomycin, a last-line antibiotic. And guess what? Vancomycin-resistant Staph aureus has been seen. Not a lot. 13 cases in 4 states over the past, since 2002. So, 13 cases in 15 years, you think that's not much of a big deal. But think about this. There's nothing you can do for those patients. That's it. There's nothing. There's nothing you can use. There are some brand new ones in trials or just approved that you could probably use daptomycin, but vancomycin is a very last line defense, and so the fact that there's only 13 cases in 15 years, nonetheless the CDC still says this is a concerning level of threat, because in another 15 years it might not be 13 cases, it might be 1,300 or 13,000. Well, it doesn't respond to the arsenal of antibiotics that we have. I mean, as this gentleman pointed out, you can boost the immune system in other ways. Oh, I'm sorry, I'm supposed to be repeating the questions. So the question is, so you can do nothing for these people. And my answer is, this is the last line antibiotic. There are some brand new ones just coming online you could try. There are ways to boost the immune system, but these are pretty sick patients, and it's about a 50% death rate. So they can survive.
So people who do survive, their own bodies heal
Eventually clear it. Yes.
And can those people be studied, the people who survived it, and see what bacteria they have?
You certainly can. Again, it's Staph aureus, so the antibodies you have against the bacteria would really be no different than it would. What makes it vancomycin resistant is a slight change in the cell wall, but your immune system would not be able to directly interact with that, the way the cell wall is cross-linked. But you certainly could study those patients, but I mean, even Ebola isn't 100% fatal. There's always going to be some people who have a mild case or who can survive, and that's what happened in the patients here that survived. So I've thrown a lot of acronyms at you. I just wanted to talk about some of them. So MRSA, as we talked about, this is the one you see most often in the news, is methicillin-resistant Staph aureus. MRSE is methicillin-resistant Staphylococcus epidermidis, which is just another type of staph usually found on your skin, but it can cause serious disease. As I said, for MRSA, you have to use vancomycin, so you start to see VISA, which is vancomycin intermediate resistance, meaning you have to use a very high dose of vancomycin. And then when even that doesn't work, it becomes VRSA, which is vancomycin resistant Staph aureus, those 13 patients. But the big concern is CA MRSA. Has anyone heard of CA MRSA? Correct, community acquired. So who usually gets these infections? In CA MRSA, yes. So for most bacterial infections, not always, but usually it's very young, infants, babies, toddlers, people that don't have very strong immune systems. It's elderly, people who have other health problems, okay? It's people who are immunocompromised, who are taking immunosuppressant drugs for cancer, maybe they got some type of organ transplant, or just have an otherwise weak immune system. These are the people who you usually see in the hospitals who have these bad infections. And to many of the people here, you're thinking we're 15 years old, we're 20 years old, college students, robust immune system, we are the picture of health. You are the ones, and no one knows why, who are susceptible to community-acquired MRSA. Okay, community-acquired MRSA, you get it in groups. It's huge issues in locker rooms, in gymnasiums, and it mainly affects young, healthy people with robust immune systems. No one really knows why. There are some theories, but I don't want to get into a long sidetrack about those. But it affects everyone. In fact, we have a professor next door at my institute, Zvi Kelman. He's late 40s, 50 years old. He had a bump on his arm. It was a little red. 2 days later, he was in the hospital. He was there for a week. He had to go on. It was methicillin-resistant Staph aureus, community acquired. He is still on. He has been on for 30 days, daily IV vancomycin. And he's in perfect health. And it just showed up as like a boil on his forearm one day. So this is really scary, and this is usually what you see. So I put this together yesterday, and I've just confined this to 2017. We're still in March. District officials confirmed a case of MRSA at Springdale High School. This is in Springdale, Pennsylvania, in January 18th. Falkirk Times, high school athletic departments wrestled disinfection issues. This is January 26th. This is right here in Virginia. They had outbreaks of MRSA in the wrestling team, and they're out bleaching the wrestling mats. Same day, January 26th, MRSA infection found at a youth center in Westfield, Massachusetts. Close the center. Where are we now? We are now February 1st. Buchanan High School in Buchanan, Michigan alerted students may have MRSA, closed down the school for disinfection and cleaning. And what did we learn from NIH? Disinfection and cleaning doesn't necessarily do the job. Here's in Southampton, New York, February 3rd, MRSA infection at Southampton High School. The school will undergo a deep clean this weekend as a precaution. Here we are, March 27th. I said 3 days ago. Bedford cafeteria worker diagnosed with MRSA. This is in Bedford, New Hampshire, closed 2 schools. So these are the headlines that you see every day because of MRSA, and most of this is the community-acquired MRSA. Does anyone know who this is? This is Tynes. I forget his first name. He is a place kicker or was a place kicker for the Tampa Bay Buccaneers. He's an NFL player. And one day, just like my friend next door, he had a boil on his arm, and it turned into MRSA. And here he is getting IV vancomycin antibiotics. In fact, 3 Tampa Bay Buccaneer players had it. This is 3 years ago. To where you have ESPN sending out tweets that depending on what MRSA containment report says, the NFL Players Association could advise the Bucs Eagles not to play this weekend. They're going to cancel NFL games over this. And in fact, when the Bucs came to Atlanta and played the Atlanta Falcons, these are the pictures the next day of crews cleaning out the locker rooms with bleach. And in fact, the Buccaneers kept the MRSA a secret just so it wouldn't blow up on them. And of course, these things always do. And then this is last month, 3 years later. This is February 21st, 2017. His name's Lawrence. Lawrence Tynes reaches a settlement in the MRSA lawsuit. And it says he was one of 3 players, but it says here that he was seeking $20 million and claimed the infection ended his career. He never played another game after his bout with infection, and he said to this day he still has issues that have left him in daily pain. So of course they don't disclose the terms of the settlement, but one could figure it is in the $1 million range. So that's MRSA. This is the one that you usually see, the highlights and close the schools about. So that then begs the question: what is being done about this? What can be done? What are scientists doing? What are legislators doing? Well, let's start with the farm. You heard me say that 80% of all antibiotics are used on farms rather than humans. The question is why? Well, antibiotics aren't called antibiotics in the agricultural communities. They're called growth promoters. It's very well known that if you give, for example, a chicken antibiotics, the chicken becomes larger. It's not exactly known why. Again, there are some theories that it quells some of the bacteria in the gut, and the bacteria competes for the food for the chicken. So if you have less bacteria, you can put on more muscle mass. There are some things that it just decreases inflammation, and so the chicken can take in the nutrition more efficiently. There are theories, again, I don't want to get into them, but what is definitely known is you give a chicken an antibiotic, and that chicken's going to be 20% larger than if you did not. So this is why antibiotics are used in the food industry. I can't vouch for the validity of this. I did find this online. There is no reference. A 1950s chicken and a 2008 chicken. This does look a little doctored, I will admit. But when I was a graduate student in Athens, Georgia, I have a friend who had a chicken coop, and he said, sure enough, his parents say that chickens nowadays are twice the size as they were 30, 40 years ago. So let's get to the antibiotics. 7 million pounds of antibiotics are used in the U.S. by humans, and this number has been stable. This graph goes, what does it say there? This is from 2001 to 2011. So we consume the same amount of antibiotics. This is agriculture, 30 million, is it pounds or tons? Pounds, 30 million pounds. And so 30 million to 7 million, it's 80% of all antibiotics go into farm animals and not humans. So what's happened about this? Well, there's been some legislation. In 2012, the FDA was successful in banning the use of cephalosporins for growth promotion in certain livestock. So the problem is it's the same antibiotics, right? And so all the spreading of the antibiotic-resistant genes is happening in the farm animals. So there are antibiotics that do work better in animals and humans, and because they have different types of infections in some cases, but let's take MRSA. It's the same bacteria. So the same bacteria that causes MRSA in a human is the same one that causes bovine mastitis, an infection of the udder in milking cows. And so the overlap of the human and the animal antibiotics, you have about a 70% overlap. So some of the ones that are most critical, they're trying to legislate them out of use in animal feed. And so in 2012, the FDA was successful in one. There are a couple of bills in Congress. A wider ban on antibiotic use for animal growth promotion is in something called the Preservation of Antibiotics for Medical Treatment Act. Another new legislation is on delivering antibiotic transparency in animals that would require reporting of the types and amounts of antibiotics put in feed. These are still stuck in legislation. They aren't laws, but it was enough to scare the companies. So seeing that it was going to be legislated out, the pharmaceutical companies and the big agricultural farm animal companies went ahead and voluntarily restricted themselves. So this was in 2014. Almost all drug companies have agreed to go along with veterinary plan to stop the use of antibiotics to fatten farm animals, according to the FDA. 25 of 26 companies that the FDA appealed to actually said that they would change the labels and disallow the use of antibiotics. So it's slow, but it is, from public pressure, it is starting to happen in the farm animals. And I just discovered this like an hour before I came here in Maryland. This is March 21st, 2017, a week ago. The Maryland House and Senate passed a bill to keep livestock free of antibiotics. The Senate and House of Delegates here in Maryland passed Keep Antibiotic Effective Act and separate actions last week. If approved by the governor, so it's not approved yet, but if approved, it would make Maryland the second state to have such laws, the first state being California that passed this legislation about 5 years ago. So this is very, very current, just happened a week ago. So let's get back to the problem with the antibiotics themselves. You know, what's the government doing? Well, in 2014, the White House came out with the paper on the left here called A National Strategy for Combating Antibiotic-Resistant Bacteria. You can Google this, you can download, it's about a 120-page PDF. So they came out in 2014 with a national strategy, and then the next year, in 2015, they came out with a national action plan. And this is, I think, a 180-page PDF. But if you read through these documents, the overall take-home message is that there's a need for some type of disruptive anti-infection technology, which are alternatives. So new antibiotic discovery is going on, and new antibiotics will be discovered, but they're looking at alternatives, such as boosting your immune system through other mechanisms. So what are some alternatives to antibiotics? Well, there are vaccines, but vaccines you use to prevent infection. Once you have infection, a vaccine doesn't help. So it's not really an alternative. It's just a different way to prevent infection. But there's photodynamic therapy, which people are working on, light therapy, if you will, quorum sensing inhibitors. Does anyone know what quorum sensing is? My microbiologist here doesn't know? Okay. So bacteria are like a bunch of teenagers. They like to talk to each other. Bacteria are very social, okay? And we can talk to well, we used to talk to each other, now we Snapchat each other. But bacteria talk to each other through chemical molecules. And this is quorum sensing. So they send out these chemical signals and say, hey, this is a good place to grow. They'll sync their growth together. They'll do things like form these biofilms where they say, okay, nutrients are poor here. Let's kind of hibernate like a bear and slow down our metabolism. Let's spread. So they can talk through these chemical messaging system. And so a quorum sensing inhibitor is something that blocks that messaging. And then it makes the bacteria more susceptible to, you know, your immune cells, to your white blood cells, to your neutrophils, for example. It certainly could, but different strains and species have different types of chemical signals. But yes, it certainly could. Antimicrobial peptides. So these are small peptides, chains of amino acids that act on the bacteria and can inhibit growth or kill. We have these. Our saliva is full of antimicrobial peptides. Our tears are full of them. We have them in our blood. You know, you hear that the dog's mouth is cleaner than your mouth. Dogs have lots of these peptides in their saliva. This is in our blood. Our neutrophils have these. So there are people who are trying to, you know, mass produce these and use them as therapeutics. Iron chelators. Bacteria need iron to grow. So our body, we need iron for our red blood cells, but our body keeps iron under very tight control. There's very small amounts of free iron in your blood because the bacteria tries to sequester it. So you can add proteins that will do will sequester additional iron, and so the bacteria doesn't have it. There are some very advanced ways to silence the RNA. There are now things, I won't go into, is based on truth, and there are scientific publications that do support some medicinal and antimicrobial properties of some of these natural products. And then at the bottom here, bacteriophage therapy and enzymes produced by them called endolysins. And this, right now I haven't talked about my own work, but this is work that I do. And in fact, if you go into these documents here in the National Action Plan, they talk about alternatives to antimicrobials, and they list 7 ways that non-traditional therapeutic strategies should work. Targeting virulence factors, vaccines, creating therapeutic products of monoclonal antibodies or polyclonal antibodies, boosting the immune system. But one of them is using bacteriophage and bacteriophage lysins to kill specific bacteria while preserving the microbiota, meaning kill the bad, don't hurt the good bacteria. And so I'm not going to go into in depth in my research, but I'm going to give you 3 or 4 slides just so you get an idea of what we do in the lab and how it fits into this whole talk and this whole antimicrobial resistance. So does anyone know what a bacteriophage is? Yes? Exactly. So a bacteria is a virus, a bacteriophage is a virus specific for bacteria. So we have viruses that make us sick, but our cats and dogs don't catch our viruses. Our cats and dogs have viruses, but we don't get their viruses, right? So viruses act in species-specific ways, doing nothing to you, but they can kill bacteria. So this is a little flash animation of a bacterial cell. And here's the bacteriophage, and you described it exactly, like a little lunar lander. So the bacteriophage will attach to the bacteria, inject its DNA, and it takes over the bacterial machinery, if you will, to make new bacteriophage particles. Now, late in this replication cycle, as the new particles are being formed, you have an enzyme that forms a hole in the bacterial membrane, and then you have another enzyme called a lysin that goes through that hole, and it chews up the cell wall. Now, this cell wall is the rigid structure that keeps the cell intact. Bacterial cells are under 10 to 20 atmospheres of pressure. So think of that cell as a champagne bottle that's been shaken up, and think of that cell wall as the cork. So these enzymes drill a hole in the cork. So the whole thing explodes. Obviously, that bacteria is now dead. These phage particles can now go infect a new bacterium. So bacteriophage were discovered in 1917, and there was a lot of interest in using them to kill bacteria, and it's called phage therapy. So it has some advantages. You put one phage in, you get 100 out. It's very specific, so I can use a staph phage, and it will only kill staph, and it won't touch anything else. So you can target the bad pathogenic bacteria and not harm the good bacteria. So there's lots of advantages. There are some disadvantages. Phage can carry some toxin genes. You develop resistance to phage, so you need a cocktail. But a lot of people were interested in phage therapy in the 1920s and 30s. In fact, Eli Lilly, a US pharmaceutical company, sold phage preparations, and then penicillin was discovered. And then they stopped their bacteriophage research and focused all on penicillin and antibiotics. But phage research continued in Eastern Europe and in the former Soviet Union, in the Georgia Republic, in Tbilisi, Georgia, and still to this day they do human phage therapy work out there. And now with this antibiotic-resistant crisis that hopefully I've convinced you, we shouldn't be panicked, but we should be concerned. There are a lot of companies here in the US that are going back to phage therapy as a viable treatment option. So that's phage therapy. My lab doesn't really do phage therapy. We just look at these lysins that were called lysin or endolysin. Now we produce them recombinantly. We can manufacture them, grow them up, and then you just take these enzymes, so no bacteriophage, just an enzyme, and you add it to a susceptible bacterial cell. It will drill a hole in that cell wall, and although that membrane's intact, it can't hold back the internal pressure. You get this lysis event. Just like the phage, these enzymes are specific. We have a bacillus one that only works on bacillus, a listeria one that only works on listeria, and so on and so forth. Yes. So the idea of probiotics. Oh, repeat. So the question was about probiotics. Okay, can you take probiotics? So as I said, in our gut we have lots of good bacteria and we have some bad bacteria. But the good bacteria keeps the bad bacteria in check. Okay, they outgrow it. They can outcompete the bad bacteria. Now, if you kill the good bacteria, the bad bacteria will take over. So the whole idea of probiotics is that you have lots of good bacteria. Even if you have an infection with the bad bacteria, the good bacteria can outcompete for that ecological niche. And so there are a lot of probiotic ideas and treatments. You can read about it. There's something called the fecal mouth transplant, where people who, for whatever reason, some people just can't culture the good bacteria very well. And so they have a lot of the bad bacteria. So you actually can take bacteria from the feces of a relative, and they, through a tube, can put it into your intestines, and it will recolonize, and you can crowd out the bad bacteria. So I'm not talking much about probiotics, but that certainly, add that to the list of alternatives. It's definitely a very valid idea. So that's what we do with these enzymes. And as I said, these enzymes are specific in a species-specific manner. And in fact, here this is a cross-section electron micrograph of a pneumococcal cell. And our enzyme is active here on the surface. and you can see it's drilled a hole down, and here are the contents of the cell spilling out. So this is the exact instant of cell death. And then I'm going to turn down the lights for this video. In each test tube here, let me start it. Oop. So here in each test tube, we have 10 to the 7th group A strep, enough to infect everyone in this room. We added some of our enzyme to the one on the left. This enzyme just happens to be called Ply-C. It takes about 15 seconds to bind to the bacterial surface and begin to drill down. But as it starts drilling down, the cells start to pop and explode. And the test tube on the left, after about 20 seconds, goes clear. So that is completely sterile right now. Not a single bacteria living. That's how fast some of these enzymes work. This happens to be our fastest enzyme, but that's how fast they work. This isn't like a 3-hour video I compressed to 20 seconds. That's 20 seconds in real time, how these enzymes work. Here's another enzyme called Ply-G. Here we're looking under a microscope at Bacillus. So Bacillus cereus, Bacillus anthracis is anthrax, and bacilli are these long rod-like structures. And so we're looking under the microscope and we add just a little bit of the enzyme to the side, and it just kind of flows in and then starts lysing the bacteria. I liken it to like a needle popping a balloon. So you can just see the bacteria explode as these enzymes touch their surface. Okay, so those are the videos. So I'm not going to go into great detail, especially because it's 6:26. But over the past 10 or 15 years, my lab, and not just my lab, there are many other labs interested in this technology, have done in vitro studies, meaning in a test tube. We've done in vivo studies, meaning in animal models of infection. We've looked to see if you develop resistance to these enzymes, and we haven't seen it yet. That doesn't mean there will never be resistance, but so far we haven't seen it. We've looked to see if the body develops antibodies against these enzymes, if these antibodies neutralize the enzymes. We've done some studies in animals looking at toxicity. Are these enzymes safe? Pharmacokinetics is a big fancy word that means clearance. So when you take a drug, how long is it in your body? How long is it in your bloodstream? How fast do you clear it? Do you clear it through your liver? Do you clear it through your kidneys? What are the breakdown metabolite products of it? Synergy is where you can use multiple enzymes, or you can use enzymes in combination with antibiotics. So maybe you need less antibiotic, or something was resistant to an antibiotic, but in the presence of one of these enzymes, it now may be sensitive. You know, formulation, do you put these in a capsule? Do you inhale them into the lungs? Do they go into the blood? And delivery systems. So there's been a lot of work on that. There are several companies working on these. I'm aware of one. I'm not part of the company. I don't own any stock in the company, but there is a company I'm aware of, and they have licensed some technology from myself and many others. They went public and raised $140 million, and they have one enzyme that works on MRSA. And there are, Sarah, you must have 20 enzymes that work on, 15 enzymes, 50. Okay, there are 50 enzymes that work on MRSA. So this is one enzyme for one bacteria, and they raised $140 million. That's how much people are worried about antibiotic-resistant bacteria. This was in 2014. They did phase 1 clinical trials in 2015 and 16. That's safety trials, and now in 2017 they're enrolling patients in phase 2 clinical trials. So I think you're going to see more biotechs, more pharmaceutical companies start to perk up to some of these type of technologies. And this is just a list of bacteria for which these enzymes have been cloned, expressed, and characterized, some in my lab, some in other labs. But you can see here a lot of those pathogens we discussed, Clostridium, Enterococcus, Staphylococcus, and many different Streptococcus is what we're working on. To save time, I'm not going to go through these other slides. I'm happy if you want to stay. Just about some of the work in our lab. I just go over some of the different projects of some of the different people in the lab. But I will, I will go over this one. Athena, who's here in the audience, is a postdoc in my lab, and she has enzymes that work against Clostridium difficile. These are the ones in your intestine. The problem is getting this bacteria, or getting this treatment into your intestine, because the acid of your stomach will destroy it. So we have two different approaches. One, we can put it in a probiotic that will naturally colonize the intestine and have this probiotic pump out our enzyme. And the other one is we can put some type of acid-stable coating on it. You know, when you take aspirin, baby aspirin, it has enteric coating. All enteric coating is a special polymer that is a solid at low pH, so the acid of your stomach, and then when it gets into your intestines and the pH rises, the coating melts. And the drug is then delivered to your intestines. So that's just one project where we're doing probiotic engineering. We have diagnostic projects and others. Okay, I want to end now with, and feel free to ask more antibiotic questions, but I want to talk about my journey, because this is what a lot of the students want to know about, not me in particular, but you know, graduate school and the STEM programs and so on and so forth. So my journey, and I'll make this quick, begins in Milwaukee, Wisconsin, where I was born. And then when I was about 3, I moved to Atlanta, Georgia, and I went to elementary school, middle school, and high school in Atlanta, and then I started college at Georgia Tech, and I was a biology major, but I was always interested in medicine. In fact, when I was in middle school, I did the Boy Scouts, and I was a medical explorer for the Boy Scouts who provide the medical support for all the big campouts and jamborees. When I was a junior in high school, I started taking classes to be an emergency medical technician. And when I turned 8, because you have to be 18 to be one, I had already done the courses, so on my 18th birthday I took the exam and I was an EMT, working all night long on a 911 ambulance, doing shootings and stabbings, and I was still a senior in high school. I don't know how my parents allowed it. But when I went to Georgia Tech, I continued working on the ambulance, and I was really thinking about medical school. But then after my first year, I was living in Atlanta, I had the same friends, the same family, and I just wanted to go out and explore. And specifically, this is going to embarrass my daughter, I wanted to surf. So I packed up my bags, and I moved to California. And in California, I went to the University of California in Irvine. And if you think that University of Maryland, the turtle, the terrapins, is kind of a not so wonderful mascot, try being an anteater at the University of California, Irvine. So when I was at University of California, Irvine, all of the EMTs and paramedics were firemen, and I would have had to have dropped out of college to go to the fire academy, and I didn't want to do that, but I still wanted to use my paramedic skills, so I worked in an emergency room. So I was a trauma technician in an emergency room, and this is how I worked my way through college. And I also worked in a research lab, in a biochemistry lab, and I liked that a lot. And I graduated in 1993, and then I had a decision. And I know for many of you in this room, how many of you are in middle school? Okay, and how many of you are in high school? And how many of you are undergrads here? Right? So it's about 1/3, 1/3, 1/3. But obviously those in high school and middle school are interested in science, math, and engineering. That's why you're here. Those of you in college are trying to figure out what's next. So for me, I had a decision. Do I want to go to medical school, or do I want to go to graduate school and get a master's or maybe a PhD? For me, politics played a role, because in 1993, this is probably too small for you to read, there was a new president, Bill Clinton, and his wife Hillary Clinton was going to redo our healthcare system. I know everyone knows about Obamacare, but 23 years ago, an earlier version was called Hillary Care. And in fact, it was going to somewhat socialize medicine. And at the time, we had a lot of doctors. There was a glut of doctors. And so there was talk of medical schools limiting the number of applicants. And it wasn't so clear that going to med school was going to be the best option at the time for me, again in 1993. And so I thought more, well, maybe I'll go to graduate school. And I can always drop out of graduate school and go to medical school if things change, but let's try this research. So I packed my bags, drove cross country again, back to Georgia, where I was from. And this time I went to the University of Georgia in Athens, who have a good mascot, the Bulldogs. And I enrolled in a PhD program in biochemistry and molecular biology. The first year of a PhD program, you pretty much just take coursework. And then your second year, you get in a lab, and you start doing work, and then you take something called your oral exams, or your qualifying exams, where they rigorously grill you on different topics. And once you pass that, you advance to PhD candidacy. Now, you still have 3 years left, because a PhD is about 5 years. But once you pass those exams, it's downhill. You do your research and publish your papers. All this, the coursework is done, the stress of passing those exams is done. So at that point, I'm like, it's downhill. The last thing I want to do is go to medical school. I like this research, so let's keep doing it. So it took me 5 years. I got my PhD in biochemistry. And then what do you do after you get your PhD usually? Postdoc. Okay. So if you go, so medical school is 4 years, and after medical school, you do a residency and internship. A PhD can be 4 years, it can be 5, it can be 8, but it's typically around 5, and then afterwards you do a postdoc. So a postdoc and a residency for an MD are similar in that you have your degree, you have the initials after your name, you're a doctor, but you're still kind of training and specializing in what it is you do. And you get paid a little more money. You don't get paid a lot, but you get, you have a living salary. And that postdoc or that residency can be 2 or 3 years, or it can be much longer. So if you want to specialize in neurology, be a neurosurgeon, you're probably doing a 6, 7, 8-year residency and internship. Depending on what you want to do in science, you can do a 2, 3 year. I ended up doing like a 6 or 7 year postdoc. And where I did that was, let's go forward, not backwards. I did that at Rockefeller University in New York. I chose Rockefeller for a couple of reasons. Number one, it's world famous in the biomedical sciences. They have a lot of Nobel laureates. But going back to my surfing thing, you got to go somewhere fun. And Rockefeller is right in the middle of Manhattan in New York City. It's in the Upper East Side. And for those of you that have never been up there, this is the Upper East Side of Manhattan. There's Central Park. And it's in a very rich biomedical center. This is Cornell Medical School. This is Memorial Sloan Kettering Cancer Institute. And this is Rockefeller University. So the 3 institutions kind of work together and do really great biomedical science. My lab was in this building, and that was my apartment. I was actually on the 20th floor of this building looking down the East River. So it was a great environment. It was a lot of fun living in New York, but we did great research, and everything I presented to you talking about some of these endolicense, all this work started there. And in fact, they still are working on it up there, and now I'm working on it here in Maryland. Now, one other thing I did while I was in New York as a postdoc is I went to night school and I got my MBA in finance, which doesn't seem like something I'm not really molecular, a biochemist would do. But you have to realize the time. This is in 2001, for those of you that remember it. The stock market was going through the roof. This was the big technology bubble. Companies were going public and raising hundreds of millions of dollars overnight. And it was a very exciting time to be in New York in finance in the biotech arena. And so I did get my MBA. And then I had a decision. I almost became a financial analyst on Wall Street looking at biotech stocks, but then I got a job offer here in Maryland. And I came here in 2007, so I've been here for almost a decade. And people say, well, you're wasting your MBA. You aren't using it at all. But I disagree. An MBA is to be a business person. And you have to learn finance. Well, I have millions of dollars of grants, and I have to budget and finance my lab. In an MBA, you learn marketing. Well, marketing is what I'm doing here. I'm standing in front of a crowd, public speaking, talking about my research, convincing you how wonderful it is. Okay? You have human resource skills, management skills. I manage a lab. I have to manage department chairs. I have to manage different committees that review my grants. So running a research lab in academia is very much like being a small business person, running your own business. So I find that the MBA skills are almost more valuable to me in my lab than than my PhD skills, because science moves so fast. What I learned in my PhD is my students know so much more than I do about many of the things we do. So never forget that to be well-grounded is good. So I only have 2 or 3 more slides left, but I want to talk a little bit about what it is to get a PhD. Okay? Does anyone realize that if you get a PhD in the sciences and engineering, biochemistry, etc., that you actually get paid? Do you guys know that? You get a stipend. Well, that stipend is salary. It goes in your bank account. So let's use, so right here, this is Dr. Sarah Linden. She was my student. She graduated last year from the University of Maryland in the Molecular and Cell Biology program. So let's use her as an example. So the salary is about, the stipend is about $30,000 a year. It's actually $28,000 or $29,000 right now, but by next year it's going to be $30,000. So as a student, you're getting paid $30,000 a year to come and do research in the lab. Listen, there's tuition. Your tuition's waived. It's $10,000. I pay that off of my grant. I pay your $30,000 salary off of my grant. I pay your tuition off of my grant. What are you responsible for? Like a couple $100 activity fee? Yeah. And that's it. Okay. And then, as an employee of the University of Maryland, you get fringe benefits. You get medical insurance, dental insurance, pharmacy, accidental death and dismemberment, and life insurance. So you pay a small amount, but I pay 10 times more than that amount, subsidized through my grants. It's about $10,000 in yearly fringe benefits. So when you add it up to be a PhD student, your take home is $30,000, but your total compensation package is about $50,000 a year that I pay as the PI off of my grant. And I said the average student's there for 5 years. That's $250,000. Did you know that? So so people have a misconception. They're like, oh, graduate school's easier. If I don't get into med school, I'll go to graduate school. Not so. It's even tougher. If I'm spending $250,000 on someone, they have to be really good or they get kicked out. It's actually more competitive sometimes than medical school to get in. So let's look at what people with a bachelor's degree make. So the average bachelor's degree, this data is taken from the Michigan State University, and I think it's 2 years old. For all majors, the average starting salary with no experience, just a bachelor's degree, is around $39,000. Obviously, some of the engineers and software designers make a little more than that, in the mid-50s. If you get a bachelor's in chemistry, you're about $43,000. Biology, it's a little lower, around $38,000. So the point is, if you graduate with a degree in biology and you go to work in a lab as a technician, you're going to make about $40,000 a year. If you go to grad school, you're getting a $50,000 a year compensation package and a degree after your name. So that's something that I don't think a lot of people in high school, well, in undergrad, and since it's further out in high school, think about, but that's a lot of money. So let's talk medical school. And I don't want to talk anyone out of medical school. We need more doctors. It's great, but it's a very different story. So these numbers are current. These are this year numbers. The tuition at the University of Maryland is $33,000 in-state and almost $59,000 if you're out of state. That's just your tuition, okay? You also have to live. You have to have insurance that you have to pay out of pocket. Presumably you have an apartment. You have clothes. So they calculate the cost of attendance, which is about $20,000 to $25,000 more than the tuition, because you're going to have to either take out a loan from the bank, or if you're lucky, get from your parents. About $62,000 a year is the cost of attendance per year as an in-state student at the University of Maryland. Of course, if you're out of state, those numbers go up to around $90,000 a year. Let's say you want to go over to Virginia. I'm assuming everyone here is probably a Maryland resident. So to go to University of Virginia School of Medicine is $54,000 a year as an out-of-state student. Once you add in your room and board and insurance and incidental expenses, it's about $78,000. So it's really not cheaper to go to a public school than it is to go to a private school. If you go to Georgetown, the tuition is $53,000. And Hopkins, one of the best medical schools in the country, is actually a bargain if you consider $47,000 a year a bargain. So if you look at these numbers and the cost of attendance, 60, 90,000, 78, 80, 72, you're averaging about 75 grand a year for 4 years. That's $300,000 in debt. So the math becomes real simple if you're on that border like I was in 1993. With a PhD, there are PhDs that make very little money. There are PhDs that make a lot. There are MDs that make very little. There are MDs that make a lot. In general, over a lifetime, yes, it is true. A medical doctor probably does make more than a PhD, but you're starting out at a $500,000 difference. So that's something that I don't think a lot of undergrads put a lot of thought into when they make these choices. Again, I'm not trying to talk anyone out of medical school. We need more people in medical school, but $500,000, that's real money. That's something to think about. All right, I have 3 slides left. This is a slide that I made if we were 25 years ago. If you get an MD, probably 99.9% of your class goes on into medicine. If you get a PhD, probably 90% go on to academia, become a professor, researcher. There's probably 10% that goes to some government lab, or obviously pharmaceutical companies need someone. But this is the path for an MD or a PhD, I'd say 25 years ago. If you ask me what this looks like now, it's a very different story. For an MD, still most MDs end up practicing, seeing patients in one way or another, but a growing number of them are going into alternative careers. And for PhDs, I would say less than 50% of all PhDs go into academia. That means more than 50% go somewhere else. True, a lot of them do go into industry, especially with all the biotech companies, but there are a lot of alternate careers. So let's look at the alternate careers here for the MDs and the PhDs. And this is my last slide. So teaching, so there's a lot of teaching that you can do. I mean, obviously college teaching, but there are a lot of PhDs and even MDs that do K through 12 teaching. There are a lot of very expensive private schools, particularly in this area, that pay very well and pride themselves on having, a PhD in physics or math, teaching at the undergraduate private school level. So teaching is always a career. Obviously, industry, and by industry, yes, I mean pharmaceutical and biotech, but there are huge, multi-billion-dollar agricultural companies, the Monsantos of the world, the plant seed companies, and then diagnostics. Everything is getting into personalized medicine and tremendous investments in diagnostics. So there are diagnostic companies that are gobbling up PhDs and MDs. Obviously, you could go into research at a government agency, NIH, just in this area, NIH. Our institute is actually a co-institute with NIST, the National Institute of Standards and Technology. There are probably 40 NIST PhDs just a couple hundred yards away at my research institute. The FDA, so the FDA approves drugs, but they also have robust research programs. The people who approve the drugs also have their own research labs to be on the cutting edge. The USDA is located, well, there's many USDAs all over, but the main headquarters of the U.S. Department of Agriculture is in Beltsville, Maryland. They own thousands of acres up there. They do a tremendous amount of research. The Department of Energy, RARE is the Walter Reed Army Institute of Research. So they're very focused in military medicine. Funny enough, a lot of my funding comes from the Department of Defense because the soldiers with advances in body armor, when they get shot or when there's a bomb that blows up, they may lose an arm or a leg, but because of the body armor, they survive. And then they end up back at Walter Reed or at Fort Sam Houston in San Antonio or in San Diego, and then they again go through the next year or so of reconstructive surgery, getting prosthetic limbs, and they fight these horrible bone infections. So the problem is the bacteria, the multi-drug resistant bacteria. More money is currently being spent by the Department of Defense. I don't know if it's more, but from what I've seen, a lot more funding opportunities have come up through the Department of Defense than even through NIH for antibiotic resistance and antimicrobial research. Obviously the CDC. So these are all government agencies where you can do research as an MD or a PhD, and they especially like MD PhDs. If you're a real glutton for punishment, those people do very well. You can be an analyst or a program officer. So I write grants to the DoD, to NIH, to NSF, and they have PhDs and MDs who are the program officers who manage their grant portfolios, who decide where the next scientific priorities for funding are. DARPA is part of the military. It's the Defense Advanced Research Project Agency. They're full of PhDs and MDs interested in biomedical research, but they're more as a program officer or an analyst. Even the State Department, the FBI, hires a lot of PhDs and MDs. And then policy, Congress and Senate, State Department, US AID, the US Agency for International Development. A couple of weeks ago, I was on a review panel for all of these American Association for the Advancement of Science fellowships. They take new PhDs who apply for these fellowships, and you go work for the State Department or USAID, or you go work for a congressman or a senator as their scientific policy advisor. So, you know, there's always these scientific bills that some senator puts forth. Well, that senator isn't a scientist. He or she has scientific policy analysts that advise them on this. So there's a tremendous amount of policy that you can do with a PhD or an MD. Management consulting, this is an interesting one. So I've listed here the big, the big 4 or 5 management consulting firms. They recruit right out of PhDs, not because of your great knowledge in biochemistry or engineering, but it's in your mind and how you think. And the best way to explain this is, I have a postdoc. I don't have a technician currently, but let's say I have a technician, and I have a postdoc. I tell that technician, I need you to go do this experiment, and that technician does the experiment, does it better than I could do. But if there's a problem, the technician comes back to me and says, it didn't work. And then I have to be, well, did you try this? Did you try that? Well, maybe do this next time, right? The postdoc does the experiment, and it fails. And then she does it again, and it fails. And then she reads online and figures out what issues other people have had, and then decides to change some of the buffer conditions or change the incubation time, tries 5 or 6 things to troubleshoot, and then comes to me and either says, it didn't work, and I did X, Y, and Z, and this finally solved the problem. That's the difference between the technician and the PhD. It's that you have the analytical skills to solve the problem. And it doesn't matter if you got your PhD in fruit fly genetics or in studying malaria or studying cancer. It's that you spent those 5 intensive years of studying and thinking outside of the box and troubleshooting and coming up with solutions, and that's what the consulting agencies want. They don't care what you did your PhD in. If you got a PhD, you can troubleshoot, you can come up with alternatives, you can bring solutions, not problems. I say that a lot. Bring me solutions, not problems, okay? So if you have a PhD or an MD, you're very, very valuable to these large management consulting companies that pay 6 figures straight out of your PhD with no postdoc or anything. So that's a very active area. Intellectual property, patents. So, I gave you some examples today about some technology we're developing. There's patents on this technology. There are companies interested in that technology. So I spend a lot of time sometimes with lawyers. So if you have a PhD, the U.S. Patent and Technology Office wants to hire you because wants you to be the one who can assess the technology of the patents that come in. For the same reason, the law firms who are sending those patents in want you to write the technical aspects of that. Okay? Pharmaceutical and biotech companies want you for that technical background to be on their legal teams, right? To help their lawyers understand the technology, to look up the other research. And then finally, universities. At the University of Maryland, we have a legal team that must have 2 lawyers and 7 PhDs. And it's the PhDs that go out and talk to the other scientists and help you do your invention disclosure and help you file your paperwork. The lawyers have to actually, submit it to the courts and prosecute it, but it's the PhDs that do a lot of the technical writing and do some of the licensing agreements and so on. Financial analyst. So I told you I was almost a financial analyst. Venture capital firms, banking. I listed here, J.P. Morgan, Citigroup. A friend who got his PhD when I did, he is a financial analyst and makes more money in 1 year than I will in my entire life. He's highly successful. But what he does is, so he works in banking for these big firms on the sell side. So what that means is he goes to all the scientific meetings, he reads all the scientific papers. Some doctor from Johns Hopkins, some MD, PhD from Johns Hopkins just had a hot paper in Nature about this new cancer therapeutic, and that technology has been patented by Johns Hopkins and has been licensed by Merck. Okay? He reads that patent, he goes to those meetings when that scientist is giving a presentation, he sits there and asks questions, and then he comes back and writes a report about whether he thinks this is the next great gold mine for Merck or not so much. And then as a client of the bank, you pay for those reports. You pay for his advice. When he upgrades or downgrades a stock based on his knowledge of it, millions and millions of dollars are traded based on his thinking, and it's his PhD that allows him to make those calls. There are actually a lot of MDs who are financial analysts for the same reason. Journalism. So all these scientific journals, they all have editors, scientific writers, nowadays bloggers, podcasters, they're all out there spreading science. Some of this gets in with science policy, but there's a huge influx of scientific journals that all have editors who are PhDs who review and manage and do guest editorials. And then lastly, sales and technical support. We recently bought a 900 megahertz NMR, which is a large magnet to study little molecules, but it's $5 million. The salesperson had a PhD. The salesperson has to come explain the technology. We buy fancy equipment all the time. You get a lot of people with masters and PhDs and even MDs who work in sales for these very technical scientific equipment that can help troubleshoot, that can help explain it, help set it up, help develop protocols and techniques to use the instrument to the best. So again, I took time to go through this because I don't think a lot of people, when you say, oh, I want to go to medical school or I want to get a PhD, it's not a foregone conclusion that you might practice medicine or you might be, a professor somewhere. It's these alternate careers is where the future jobs are and where the people are going. So that's something to keep in mind. So with that, I will stop. Happy to take questions about this stuff, questions about antibiotic resistance. If you need to leave, you can. If you want to stay and longer, ask more, just go ahead. I've taken all the time I was allotted. Thank you. Yes.
So, what was your favorite surfing beach in Southern California?
Huntington.
Huntington.
Yes. It's definitely Huntington. I lived at Seal Beach, but I would go down to Huntington. Oh, yeah, 99. So, obviously you have a cow who's sick and needs an antibiotic and a veterinarian comes and prescribes it, yes. But 99% of that isn't from a veterinarian prescribing it, it's in the feed. The big feed companies are dumping this in by the barrelful and mixing it up. And so there's no prescription, no nothing. It just gets shipped in the feed. What's that? The animals get treated if they need it, but that's less than 1% of the antibiotic use. I would say the legitimate use of the antibiotics. It's a very small fraction, yes.
So
Certainly, yes. So we do, oh, repeat the question. So the question was, so we've talked a lot about antibiotic-resistant organisms in human disease. What about in animals? The question was specific about pets. Yes, dogs and cats can get sick, do get bacterial infections, just like us. They get viral infections just like us. I don't know that much about domestic pet animals. You know, for example, dogs do get Pseudomonas infections in their ears quite often, and a lot of times that can be multi-drug resistant. What I'm more familiar with is farm animals. So we work with bovine mastitis, so a cow that's lactating, a milking cow, can get an infection in the udder. And what happens then is that the milk quality, first of all, the cow produces like 40% less milk, and the milk that the cow does produce is of low quality, and you have to waste it. And so it's this huge economic impact. That can be caused by Strep uberis or Staph aureus. Those are two things we study. So Strep uberis only infects cows. It won't infect us. But Staph aureus, it's the same. The same Staph aureus out of a cow is the same Staph aureus that infects us. So something like that is called zoonotic. Zoonotic means infects animals and humans equally. We have another project we work on with the USDA on a bacterium called Streptococcus suis. Strep suis infects pigs, causes disease in pigs, but it can also infect pig farmers. So it's zoonotic as well because it can infect pig farmers. Another bacteria we work with is Streptococcus equi. This causes essentially strep throat in horses. Okay? It doesn't infect humans, and our strep doesn't infect the horses, but the horse has a horse-specific Streptococcus that causes this horrible strep throat, but it's so horrible that it breaks down the muscle of the horse. And if you have a $10 million thoroughbred racing horse who catches this, that's very, very serious. They'll never race again the same. Many of these animal pathogens, to answer your question, do have multiple antibiotic resistance. Much more of the diagnostics and study has been for humans for obvious reasons, but it does occur in animals as well. Claire. There have been a couple of new approvals. No, there's, for the traditional antibiotics, it's still going to be your big powerhouse Pfizers and Mercks of the world. For some of that big list of the alternatives, a lot of that's coming out of Europe. Because just the mindset in Europe is more against like genetically modified organisms and more naturopathic ways, and so they've embraced alternatives more so than the US pharmaceutical companies. Both are embracing it now, but Europe definitely has the edge in some of the alternatives, whereas most of the new traditional antibiotics are out of US companies. Yes.
Which of the alternatives do you think are the most cost efficient or reliable versus
That's a good question. I would say probably, so a lot of this stuff is pretty expensive. And, you start talking about some of the monoclonal antibody therapies, it's not going to go over well in those countries. I think the girl who was here before brought up the probiotics. I think if you can get the right combination of probiotics, that might go over well. Yes. Oh, good heavens. Now you're testing my knowledge. Amox is a beta-lactam class? Yeah. Yeah, yeah. Chances are you're lots of resistance. It's in the beta-lactam class. So you have beta-lactams, which is your basic penicillin structure, and bacteria develop very rapid resistance to that. So then they have what they call the extended spectrum beta-lactam antibiotics, where the beta-lactam ring is modified, so the enzymes that break it down don't break it down as efficiently. But now we have the extended spectrum beta-lactamase producing organisms. So yeah, there's lots of amoxicillin resistance out there, but a lot of, I mean, like I said, it's just for conventional reasons. We still call it methicillin-resistant Staph aureus, even though methicillin hasn't been prescribed in 30 years. So it just kind of defines the class. So MRSA your amoxicillin would not work on MRSA, definitely. Any other questions? All right, well, thank you very much. If you have any questions, you're too shy to ask, just come up here.